In this movie, we'll discuss the role of calcium for transmitter release from presynaptic axon terminals. Here's the bottom line. Calcium is the agent responsible for the release of transmitters at synapses. And two, the role of the invasion of an action potential into the presynaptic terminal is simply to open voltage-gated calcium channels, which then allows for the influx of calcium. So in this movie, we're going to go over some of the seminal experiments which showed that calcium is indeed responsible for transmitter release and that the role of depolarization of the axon terminal is simply to open the voltage-gated calcium channels. The synapse that was used to demonstrate these features is in our old friend, the squid. Specifically, there is a very large neuron in the brain that sends an axon to the stellate ganglion, where it makes a synaptic connection with the giant axon that runs along the length of the squid and which was used by Hodgkin and Huxley in their studies of the ionic basis of the action potential. The two neurons in this synapse are so large that electrodes can be placed in both the presynaptic axon terminal, shown in green, and the postsynaptic axon, shown in red. The exceptionally large sizes of the axons allow investigators to simultaneously record the electrical responses in the pre- and postsynaptic cells, something that is not possible to accomplish even at the neuromuscular junction. But they could also perform other manipulations. As we shall see later in the movie, investigators could block various ion channels with toxins and chemicals of one sort or another. And they could manipulate the amount of free calcium in the presynaptic terminal and visualize when it was present. The way they did this is with two types of calcium buffers, chemicals that bind any free calcium in the axoplasm. One type of calcium buffer is injected into the presynaptic axon at high concentrations and binds all or most of the calcium influx that normally would cause transmitter release. Here is an axon terminal loaded with calcium buffers. The voltage-gated calcium channels are also shown, as are two docked vesicles loaded with transmitter. The only thing the vesicles require to fuse with the presynaptic membrane and release their transmitter is to bind calcium. When calcium enters the terminal, however, the concentration of buffers is so high that the buffer literally gobbles up all of the calcium, preventing any free calcium from binding to the vesicles, which prevents transmitter release. This is the recording arrangement for the stellate ganglion in the squid. The electrode in the presynaptic axon is filled with a calcium buffer, which is injected into the axon. An action potential is then evoked in the presynaptic axon, which is seen by the recording electrode. However, there is no response in the postsynaptic cell, even though the action potential invaded the presynaptic terminal. And this shows that if calcium is bound by the buffer, an action potential does not release transmitter, and hence there is no response in the postsynaptic neuron. The second type of calcium buffer is injected at lower concentrations and is conjugated to a molecule that fluoresces when the molecule binds calcium. Since the concentration of buffer is low, the buffer can bind only some of the calcium influx, and the remainder then binds to the vesicles and causes transmitter release. With these buffers, investigators can actually see the influx of calcium, and they can record the postsynaptic response with the electrodes inserted in the postsynaptic cell. Here, some of the fluorescent buffer is going to be injected into the presynaptic axon. There it goes. After, after the fluorescent buffer is injected, an action potential is evoked in the presynaptic axon, which in turn evokes a fluorescent signal in the axon terminal. A moment later, an action potential is evoked in the postsynaptic axon. This demonstrates that when an action potential invades the axon terminal, there is an influx of calcium into the presynaptic terminal. Finally, the investigators can completely bypass an action potential. Here, the presynaptic electrode is filled with calcium chloride, and that calcium chloride is then injected into the terminal. That in turn evokes a fluorescent signal in the terminal, followed by an excitatory response in the postsynaptic cell. And this shows that calcium alone can cause the release of transmitter, and that an action potential isn't even necessary. But there is a flaw in this study, namely that injecting positive calcium into the terminal caused the terminal to depolarize. So from this experiment alone, it is not clear that depolarization in addition to calcium may be necessary for transmitter release. But the next set of studies shows conclusively that calcium is the agent and eliminates any role for depolarization except for the role it plays in opening calcium channels. In these studies, the presynaptic terminal was voltage clamped, and thus the membrane potential was not allowed to change from a preset value. The voltage-gated sodium, potassium, and calcium channels are shown, and the experiments were conducted while all of the sodium and potassium channels were blocked by bathing the preparation in a solution containing tetrodotoxin, which blocks voltage-gated sodium channels, and tetraethylammonium, which blocks voltage-gated potassium channels. The only channels that were not blocked are the voltage-gated calcium channels. Next, the presynaptic axon membrane is voltage clamped to minus 20 millivolts by the injection of positive charges into the terminal. The depolarization then opens calcium channels, allowing for the influx of calcium. Remember, if the membrane potential tries to change as calcium enters, the electrode passes an equal amount of negative charges, thereby canceling the calcium influx and clamping the membrane potential at minus 20 millivolts. Remember also that the negative charge that we inject is actually a reflection of the positive charge being carried by calcium. That is, the current that's measured is the calcium current, since the calcium channels are the only channels that are not blocked. The records obtained in this experiment are shown in the slide. The membrane was voltage clamped for only 3 milliseconds, and during that short period of depolarization, the calcium channels opened and a calcium current was recorded as shown in the middle record. A moment later, there was an excitatory potential recorded in the postsynaptic cell. What this shows is that when the cell was depolarized, the influx of calcium caused transmitter release and a response in the postsynaptic cell. But this is the definitive experiment. Here, not only were the sodium and potassium channels blocked, but the calcium channels were also blocked by the addition of cadmium to the bathing solution. Now all the voltage-gated channels were blocked, and when the presynaptic cell was depolarized to minus 20 millivolt, there was no calcium current that entered the cell, and there was no response in the postsynaptic cell. So even though the presynaptic cell was depolarized, the depolarization did not cause the release of transmitter, since the calcium influx was blocked by cadmium. This showed that when the calcium influx is blocked, there is no release of transmitter and no postsynaptic response, even though the presynaptic cell is depolarized. So we come now full circle to the conclusions. And the two are, first, the factor that is essential for transmitter release is the influx of calcium into the presynaptic cell. Two, the depolarization itself does not cause the release of transmitter, but only acts to open voltage-gated calcium channels that then allows calcium to enter the presynaptic terminal. It's all calcium.